Good afternoon, everyone. My name is Jenny Zenobio. So today I will present about interaction between microplastics and PFAS, two emerging contaminants currently in the environment. I will start talking about a little bit of background. So we already know about a lot about PFAS and microplastics. These two are two emerging contaminants present at pretty high concentration in the environment, depending where we are looking for. They have been detected frequently in different environmental and biological matrices. PFAS are synthetic organic compounds that have been used since 1940s. They have multiple fluorine atoms, which make them hydrophobic and aliphobic and very resistant to chemical and thermal treatments. Two of the most studied PFAS are PFOA and PFOS. Microplastics are fragments of synthetic materials that their size can range from 1 micron to 5 millimeters. They have different shape, color, size, and density, and they have a pretty high absorption capacity for hydrophobic chemicals, which make them ideal for PFAS, which also become the important vector to carry toxic contaminants and facilitate the transport into different species. With that, we start to study PFAS and microplastics to see how they are interacting, how they're absorbing, and what could be the implication of that. So we did a couple of absorption tests, and we select 6 different PFAS and 5 different microplastics. For PFAS, we select PFAS with different carbon change and with different functional groups. But we go from C4 to C8 for the length of the molecular, and also we select sulfonates to compare with carboxylates. We also select fossa because fossa have C8 or 8 carbons, and we can compare their absorption capacity and behavior with PFOS. For microplastics, we select the ones that are frequently found in the environment at pretty high concentrations. We select polystyrene, PVC, LDPE, polypropylene, and PTFE. What we found is that, like with different absorbents, that higher absorption was found for sulfonates comparing with carboxylates. The lower absorption obviously was for the PFAS with shorter carbon change comparing with the C8 or fossa. The highest absorption of all the 6 PFAS selected was for fossa comparing with the other sulfonates or carboxylates. And also the lowest absorption was for microplastic was for polystyrene, and the highest for PTFE, which also have fluorine in their structure. With that, we did a couple of toxicity tests to see how this absorption of this interaction with PFAS and microplastic will affect the environment. So we select airworms, and with initial PFAS concentration of 200 nanograms per gram and initial concentration of microplastics 200 micrograms per gram. Of the 5 microplastics that we tested previously, we select polypropylene as a microplastic, and we select a couple of endpoints. Today I will present only the survival rate and also the bioaccumulation of PFAS to airworms. We also did metabolomics, which I'm not presenting today because of the time of the presentation. So what we found, so we actually have a different system. We have a control where we are not introducing microplastics or PFAS. We have another system where we only have microplastics. The next system is soil contaminated with PFAS, PFOA, PFOS, or the combination of these two. And the last system was actually PFAS absorbed to microplastics, and we contaminate the soil where we introduce the airworms. So with that, we observed that there was not so the survival rate for the system with microplastic by itself was 100%. So, but when we introduced PFAS in the system, we saw a decrease in survivability rate, which decreased in week 1. Until week 4, we have approximately 80% of survival rate when PFOA, PFOS, or the combination of these two are in the soil. And the highest effects were observed when PFAS was absorbed to microplastics, which produced a reduction in survivability rate of 50%. We saw the highest absorption was for PFOS compared with PFOA, similar what we saw in the highest bioaccumulation, similar to what we saw in the absorption test with microplastic, which means that microplastics is actually introducing the PFOS into the organism. With that, we can say that the airbornes can actually transport different contaminants into higher organisms, in this case vertebrates, and can be introduced in the food chain.
We're right at time.
And I was
Great.
just thank the team that.
Yeah, go ahead.
So I just want to thank my advisor and also my PhD students and also others that work in this project. Thank you.